Palmgren, and today I'm going to be presenting my stats final project on gene expression data analysis. So the data that I used came from GTEx portal, which contains several data sets that hold gene expression information for tens of thousands of genes with hundreds of tissue donors. With some of these samples comes publicly available metadata, which can be interjoined with the data. And so I analyzed some of that too. So I looked at two different data sets for this project. The first one is median gene expression across various tissue types. The idea with choosing this one was to try to get a rough idea of how gene expression looks across all different tissue types that are available. And then I also looked at gene expression in hippocampus tissue with subject-specific metadata. And then the goal with this one was just to see how metadata factors like age and sex correlate with gene expression and hippocampus, and trying to see if I can draw any conclusions about cognitive function from that. To start off with, I ran a PCA to look at the variation in the structure of the data. I found 40% of the variation to be explained by PC1. And so diving into that further, I made a biplot to see if there were any genes driving that variation more than others. And that is this right here. And then that led me to look at this little group right here. And so looking at the top 10 positive loading values, I found these genes to be driving that variation seen in PC1. I researched further and found 8 out of 10 of these genes to be proteins that are secreted by the pancreas, which I thought was interesting. And then the other two genes code for two subunits that make up hemoglobin, which is the red blood cell. Some additional summary statistics showed that whole blood had the least number of genes expressed, which might mean a highly specific expression profile, especially if HBB and HBA2 might be correlated to this. And then not the most expressed genes, but the greatest range of gene expression is a pancreas tissue. So this may mean a unique expression profile on top of driving variation. And then I thought it was important to note that insulin was among those top variation driving genes. And since insulin is carried throughout the blood, it makes me think that maybe those two blood cell genes being among PC1 drivers isn't like a separate thing, and that maybe they're connected somehow. So created a correlation heat map, and then this is like the very bottom corner of that correlation heat map. The correlation heat map is a lot bigger than this, but I kind of screenshotted this bottom corner because it really highlights kind of those same tissues and samples that I was getting at in the previous slides. You see pancreas tissues and then whole blood again just do not share similar gene expression profiles to other tissues. I think like if you go back and look at the entire picture, obviously I didn't have enough space on this slide, but the entire correlation heat map is bright red except for this little cross right here of blue. And it really just kind of like highlights the difference in gene expression profiles between whole blood and then pancreas, which I thought was pretty interesting. And so next, I kind of wanted to analyze the difference in gene expression profiles between sex organs. Obviously, they both serve very important roles in human reproduction and stuff like that, but they also serve completely different purposes as well. And so I really kind of wanted to take a look and see if these gene expression profiles were more similar, or if they were more different. And so I made this scatter plot, which you can see on the slide right here. You can see the identity line going through it as well. And you're not seeing too many points kind of line up on that identity line, which would points lining up on that identity line would indicate very similar expression profiles, but that's not really what you're seeing here. Mostly you're seeing this group of genes that are expressed in the testes and kind of like at a low expression value. And then like hardly any expression value in the ovaries. And then you also see a couple genes that are highly expressed in the ovaries, but almost zero expression in the testes. And so I calculated the correlation coefficient for these two, and it gave me a not like a super high value, but higher than I would have expected based on this graph. And so kind of looking at it, it kind of makes sense. You see in the very bottom corner is like a lot of low expression genes that just like aren't expressed in either the testes or the ovaries, and so I'm thinking that's kind of what's driving that correlation coefficient to be higher than it actually is. So moving on into our second data set, our hippocampus tissue. I kind of wanted to get another idea of roughly the structure of this data. So I took the most highly expressed gene from the previous data set, which is going to be our WASH7P. And I wanted to kind of see how it's expressed by age group and then how it's expressed by sex. And I'm not really expecting too much variation by either of these factors, just because it is so widely expressed in all tissues. And so starting off with WASH7P by age, you see this box plot. You do actually kind of see like this little decreasing trend going on. And so I ran linear regression, and there is significant variation in the 60 to 69 age group and then the 70 to 79 age group. But then kind of like looking at the model, it's just not a super great fit. So I wouldn't really consider that too reliable. And then looking at WASH7P by sex, these box plots, they look almost nearly identical. So no real variation there. I ran a t-test on sex, and then yeah, the p-value for that pretty high. So nothing really crazy going on there. I looked at heat shock proteins, really trying to see variation by sex, and there have been some previous studies showing that heat shock proteins, which respond to stress stimuli in the hippocampus, can vary at different expression levels based on sex hormone levels. And so I wanted to see if I could kind of see something going on there. So I looked at 15 of these HSP genes, and the only one that really showed any kind of any kind of visual difference in the box plots was this HSPE1P8. You can see the box plot there. And so I ran linear regression on this one. It didn't really determine it to be significant, and then also just not like a super great fitting model for that one either. So next I ran PCA for the top 1,000 most variable genes, and you can kind of see like a little cluster going on of similar gene expression profiles, but I also color coded the plots by sex and by age, and neither of those two factors really seem to be contributing to that cluster at all. So continuing on that, I also did some clustering with k-means and hierarchical PCA plots, and I'm not gonna lie, I'm not too confident on interpreting these really. But for k-means, it just looks like clusters are based off of variation from one another, but it's like a spectrum more than anything, so I'm not really sure how much it tells us. And then hierarchical does look like there's some degree of nested structure, but again, I don't really see anything clearly discernible in the clustering. I also made some data tables with the metadata by cluster, and nothing really stood out here either. Finally, I ran a random forest classification model on the data, trying to predict age based on expression values. Just due to our memory restrictions, I had to settle for 100 random samples and 50 random genes with 500 trees. Ideally, I'd like to get those numbers up to get a better idea of a better representation of the data. And the model was able to predict just barely over half the age groups correctly. So, not great. Again, I think with more genes, more samples, we can definitely get that predictability up. And then the variable importance plot on the right, I wanted to highlight two genes that kind of show up in both mean decrease accuracy and then mean decrease Gini, I think is the other one. But yeah, both show, I think that's CAC NG7 and then WWC2. WWC2, I think there's still some ongoing research as to what exactly this gene does, but some have found it linked to cancer in the brain. And then I think that CAC NG7 is like an ion channel that's super important for cognitive function as well. So I found those to be kind of interesting and kind of go along with the data that I've been looking at. Limitations and then areas of further study for the future. So as I mentioned earlier, limitations were definitely time and limited R memory. So with more time, I think I can definitely go in and look at other things. I wanted to originally analyze trends in mitochondrial genes versus nuclear genes. I thought that would have been interesting. And see like kind of where expression varies between those because you see blood cells, they don't necessarily have mitochondria, and so that could be a trend that I could look at. And then I also wanted to take a deeper dive into gene expression across pancreas tissue. That just seemed to be a reoccurring trend that I would really like to get into. And then also compare pancreas expression against whole blood gene expression because there's definitely something going on there. And yeah, that about sums it up. Thank you for watching.